relationship with microorganisms. Okay? Often the genetic information of the diverse microbiota except that of the house. Another is we are cooperating. It's a cooperation. Okay. Cooperation between the host and the macrobiota contributes to the fitness of the holobiome. Okay. Cooperation and symbiotic. Another one is variation. Because in the variation, the hologenome can be brought about by changes in either the host or macrobiota genome. Under environmental stress, symbiotic microbial community can change. And number four is a symbiotic microorganisms are transmitted between generations. So if we wanted to write here, the first one was a symbiotic relationship between animal, for example, or whatever plants with microorganism. Number two is cooperation. Cooperation with this microorganism which is in our body. Number three is a variation in the hologenome can be brought by change in the either host, can be about change in either host or macrobiota. And lastly is symbiotic microorganisms are transmitted in between generations. Okay. The hologenome of theory of evolution is the base that four criteria we said. They are all together, that four together suggest that the genetic wealth of diverse microbial symbionts can play an important role both in adaptation and evaluation of higher organisms. So that four can give to me the genetic wealth of diverse microbial symbionts can play important role both in adaptation and evolution. The hologenome theory contains Lamarckian aspect within the Darwinian framework, accenting both cooperation and competition within the holobiome with other holobiomes. So Human Microbiome Project started in 2007. By the way, you know that what is microbiome? When we born, we have already microbiome. So Asian Microbiome Project started in 2009. I told you why is microbiome important? What you can tell us is here, right? So microbiome is important. Why? So the genome of microorganisms that are residing in the host generally referred to microbiome. Do not forget that. So genome of microorganisms that residing in the host is microbiome. We have a core human microbiome as we see in the picture, and we also have a variable human microbiome. The variable human microbiome will change with the host genotype, host lifestyle, host pathobiology, host environment, transite community members, host immune system, and host physiology. That will eventually affect the core human microbiome also. So human microbiome you can find in the skin, in the oral cavity, urogenital tract, and the gut. But the most number of microorganisms is in the gut because this is the place that significant energy harvest from food and also immune function. How can we study the microbiome? What's the study tool? Remember, this is the metagenomics. Okay, how we will study? Because the cost-effective is a culture-independent approach to identify the macros and analyze microbial genome. It explores the genome content of the microbial community and leads to analysis of changes in the content and the explanation as a function of size and various data of their food creation. Metagenomic approach is a microbial sample are corrected from the ecological niche. I told you it will be isolated, it will be lyzed, and DNA will be purified. So let's see how we will do it. The different methods, right? In the small subunit of rRNA, just in here I want to tell you about the important about 16S rRNA. Because it's familiar for all organisms. So when I find this one, I can recognize what I am talking about, what organism we are talking about. Okay. So okay, we go to metagenomics tool. So the first one is the PCR-PGG. Okay, so we want to talk about this one. So it's done because it's one of the tools using the metagenomics after isolating DNA. So first is I will isolate the DNA. It's always remember we will isolate the DNA. What next we will do? If you recall, we will purify, second, right? Purify, and then we will amplificate. Amplificate. Okay. So after amplified through PCR, okay, and PCR product would of course be mixture of the different 16S rRNA gene of the bacteria that are found of the body or in the environment, it doesn't matter where we get it. So in order to separate the different 16R rRNA gene, PCR product is run in the gene, okay, run in the, run in the gel, because this gel is composed of denaturing, right, gradient gel electrophoresis. So we are running in the gel. The gel has a denaturing agent. Denaturing agent can be urea or formamide, and it also apply. What will happen? Those things will be separated because those hydrogen bonds will be separated. But if you look at the bottom of the test tube, it will be very dark because the more GC content, and the top will be very light. It's a less GC content. Okay. So 16S RNA denature easily, and the bound for this DNA and 16 acid would be found at the top portion of the gel, and the DNA application with higher GCC content will be the lower portion. It's doing DGGE, so what will happen? We will see the different bacterial 16S RNA will be separated. If we excite the bound and DGG and re-amplify the bound and sequence, you can be able to identify the organism. This is another isolation of DGGE. You have a different here. You have a different sample, so there will be a different migration of bonds in the gel depending on GC content and bound. So you will be able to separate genes of different bacteria in the sample and identify this bacteria in the sample. How about the real-time PCR? In real-time PCR, what we will do is useful if you want to quantify DNA, right? Quantify DNA in your sample or microbial DNA in sample. In real-time PCR, we are using the fluorescence. Okay, the fluorescence. Okay, we are using the fluorescence. And then when we use the fluorescence, and the label primer is used so that amplification of your DNA sample can be quantified. And of course, it would be the density of the fluorescence that is measured by the PCR machine, and that would be proportional to the amount and product that result from amplification. And so doing quantify in terms of fold of microbial DNA in your sample. The next one that I am really interested, this is the one, is a T restriction fragment length polymer fusion. So also involves the extraction of DNA from microbial community. Again, we are extracting, we are isolating, we are purifying, we are amplifying, doing PCR. But here we are doing PCR using fluorescently labeled 16SR RNA for 1 primer. But after amplification, there is a restriction digestion. So after amplification, amplification. Only difference here. There is restriction, digestion. Okay, as PCR product, and the fragment will be run in the sequence of the gel, and there is a software now to identify the bound. So when we get the extracted DNA from the community, what we will do, we will isolate, we will purify, and then we will do the amplification. After amplification, we are doing the restriction digestion. When we do the restriction digestion of the gel, PCR product and the fragment will be run in the sequence of the gel, and there is a software now to identify the bound. Okay, so and the last one is the pyrosequencing. Pyrosequencing is very easy. It's dependent to 1, 2, 3, 4 enzymes, the polymerase, the sulfurylase, and luciferase, and agarase. Okay, what it will does, if you remember, so it will be very straightforward. In pyrosequencing, there are enzymes involved. We said that upon addition of the nucleotide in the sequencing reaction, there will be activity of the polymerase. Activity of the polymerase will lead to formation of ATP. When there is ATP, there will be light. Okay, when it's coupled with the luciferase enzyme, there will be reduction of the light, and sequencing, it's called pyrosequencing. So for those students who didn't get it, we will start the pyrosequencing. In pyrosequencing, there are enzymes involved in the reaction. Polymerase, sulfurylase, luciferase, and agarase. This is agarase, the last one. Okay. So upon addition of the nucleotide in sequencing reaction, there will be, when we are adding the nucleotide to sequence, there will be activity of polymerase. Activity of polymerase will lead to formation of ATP. When there is ATP, will couple with the activity of luciferase, will take the light, and sequencing, that's how pyrosequencing. Okay, so after that we have similarity coefficient. We want to do data analysis. Sequence analysis will be plus basic, what? Who knows? Basic local alignment of stage 2. Phylogenetic analysis is also MECA. So metagenomic analysis of the microbiome is identification of the uncultured bacteria, reconstruction of the complete microbial genome. So why we do that? You should write reconstruction of complete microbial genome, construction of metabolic pathway of microbes, and localization of microbes through the use of fluorescence in situ. Okay, identification of uncultured bacteria. Also, development or change of the microbial community as human age from infant to adult. We also see the role of consolidation of the microbial community, and development of the new clinical tool using genomics data. It can be a research tool, a human microbial database, how the microbiome established independently and throughout the life. It can be over research. So, what's prospect? New treatment for inflammatory disease, new treatment for diabetes, metabolic disease, and similar analysis of the microbiome to children and the people, such in Africa and South America, as well as people with disease. Health and reveal the role of microbiome in maintaining health and causing disease. So this will be over discussion. So in here, I just wanted, because I told you I will tell you. I don't know if you can see. Okay, I just want to tell you about this, how this will happen. Okay, here you remember the simplified prime cap, this is a guanine, this is a adenine. How this will behave?